Good morning and welcome to Biology 102 Tea Time. I hope you've grabbed your caffeination. Today's lecture is going to extend from Mendel and give us a few new variations on Mendel. Now, this slide that I have up is just reminding you of where we were with Mendel. So Mendel lived in the 60s, so like mid 1800s. He's known as the father of modern genetics. He gave us the word gene and named this particular chemical that passed from parents to offspring. But he also gave us some laws. So you see over here, we have the law of segregation. That states that two alleles for the same trait segregate during gamete formation. We knew this already because we know that gametes only have half of our chromosomes inside of each sperm and egg. He also gave us the law of independent assortments. That law says that alleles for different traits also segregate during gamete formation. So traits for like hair color and eye color would not stick together, but they're going to sort independent of one another into sperm and eggs. Now, Mendel studied something called complete dominance. And complete dominance is where you have two alleles, so two allele options for every trait. And one of those allele options is the dominant allele, and one allele is the recessive allele. So in order to show the dominant trait, you need at least one dominant allele. In order to show the recessive trait, you have to have two copies of that recessive in order to show it. Now, Mendel is kind of interesting because Mendel studied something called complete dominance. And so it was a very simple system with these two allele options, the dominant allele that is always shown if it's present, and then the recessive allele that is hidden unless you have two copies of the recessive, and then you show it. And so this two clear-cut ways to inherit a particular trait is actually kind of rare. So complete dominance in terms of being in humans is a little bit rare. And so today I want to talk about some ways that we break Mendel's rules. And so this lecture is called Mendel's Variations, or Variations on Mendel, because these are ways that we go beyond what Mendel gave us. We extend that and into some sort of new territory. And so anything that isn't inherited outside of two clear-cut ways with this dominant and recessive allele is going to be breaking Mendel's rules in some way. So today I want to look at that. There are a lot of different ways that we break Mendel's rules, and I don't even have all of them up here. This is just going to give you a smattering of ideas about how we vary Mendel's laws and Mendel's rules. And I've divided this into two parts. So we're going to go all the way through part 5, or number 5 up here, polygenic inheritance, and then part 2 will cover the last bit of these variations. So let's get started with incomplete dominance. So incomplete dominance is the case where the heterozygote, right, that's where you have a capital letter and a lowercase letter, is an intermediate form between what Mendel would have called the dominant form and the recessive form. Over here in my diagram, I show this with snapdragons. So this is a kind of flower. If you are homozygous red, you would show this red color for your snapdragon. If you are homozygous white, we write that small r, and we would show this white color. Now, if we breed purebred reds with purebred whites, we get 100% pink, this intermediate color for the heterozygote. So you notice the heterozygote is one capital R, one lowercase r, and 100% of these heterozygote states for the snapdragons are pink. And so this is an expression that is a little different than what Mendel had. If we had Mendel, we would have red crossed with white, and all of your heterozygotes would be red. You would be hiding that white. And so this is a way to get another option into the system that is a little bit different than what Mendel said. Now, in humans, we see this with cholesterol receptors on the outside of the cell. This is an LDL, a low density lipoprotein. This is a gate that helps us take in LDL cholesterol and then process it. If you are a normal person and can take LDL into your cells and use it, then we say that you're homozygous normal for creating these gates that allow you to take in that particular kind of cholesterol and then use it. If you are a heterozygous state, then it means you make half the number of a homozygous normal person, half the number of these gates on the outside. You'll have a mild case of having too much cholesterol in your bloodstream. So it stacks up in the blood. It's not able to sort of come into that gate and then be processed and used. If you're homozygous for the inability to make these LDL receptors, then you won't make any on your cell surface, and this usually results in a severe case of having too much cholesterol inside of your bloodstream. Your cells can't process and take it in. That is where we get this word hyper and then cholesterol. So too much cholesterol in your bloodstream. So this is a case for this incomplete dominance where the heterozygous state is an intermediate state between what Mendel would have called the dominant and then the recessive, or these two other options. Another way that we break Mendel's rules is in the case of codominance. Codominance is the case where we get two alleles, like in our heterozygote, but this time those alleles are expressed equally. This happens in some speckleding cases, so in like the speckled chickens that I showed on the first slide, and some other livestock can have a speckleding pattern on them where you're showing two different alleles at once. If you have a dog with spotted gums, if you look inside of their mouths and kind of open up and see their gums, sometimes you'll see like black and pink, and those are two different colors of gums. And then if you have the heterozygote, you can see sort of both of them. So you can see this spotted gum pattern. And then we also see this in blood type in humans, which I want to talk about. Blood type is a little bit more complicated than just codominance, but one piece of that sort of complexity is a codominant pattern between blood type A and blood type B. So far we have looked at complete dominance. That's where you're heterozygote, you're going to have the dominant allele hiding the recessive. We have incomplete dominance, where the heterozygote state is an intermediate state, like those pink snapdragons, and codominance, where we have equal expression of the two alleles in our heterozygote. So a couple of different ways that we're sort of breaking Mendel's rules, or mixing it up just a little bit so that it makes it more complex than what Mendel proposed. All right, I want to talk about multiple alleles. So something else that you might have noticed is that often there are more than two options for our alleles. There are cases where you get more than two forms of a gene possible within a population. Think of things like eye color, where you have a whole bunch of different options, like you have blue and green and hazel and light brown and medium brown and dark brown. And eye color is a little bit more complicated than this, but you can see that there's all these different forms of this particular gene. Now, an example of multiple alleles that we see in humans is our blood group, so the ABO blood type. Now, what causes these blood types has to do with a sugar that is displayed on the outside surface of the red blood cell. So if you are A blood type over here on the table, then you will display a certain carbohydrate in a certain shape that we denote as A. Over here, we're showing it as little triangles, right, on the outside surface in a cartoon version, but something like this. If you are B blood type, you will display a different shape of a carbohydrate, different kind of carbohydrate that we're showing as little circles on our red blood cell. Now, when we get more than one option, so O down here at the bottom means that you do not produce any carbohydrate, right? So we have A, B, and O as our options for our like sort of phenotypes, and here's the genotypes listed over here. But what happens in multiple alleles where you throw in other possibilities, in this case we have kind of three primary possibilities, is then the alleles have to decide the inheritance pattern, and each allele grouping can be a little bit different. So in multiple alleles, in this case, we have A and B, and they are actually codominant to each other. So if you inherit A blood type or A carbohydrate from your mom and B from your dad, then you will actually display both kinds of carbohydrates on the surface of your red blood cell. So you can see over here, we have little triangles, we have little circles, etc. And so these two, A and B, would be said to be codominant to one another. However, A and B are completely dominant over O. So O is a recessive. So if you are O blood type, it means you have inherited an O from mom and an O from dad in order to display no sugars on the outside. If you look up here, if you are A and then you've inherited O, or this small letter I, then you still display A on the surface. Or if you've inherited B and then O blood type, then you would display B over here in your carbohydrates. So A and B are completely dominant over O. And so only if you are showing O, the only way to show O is to have no A and B and to have a double recessive right here. So multiple alleles is the case where you have more than two options, right? More than two forms of the gene that are possible for you. All right, the last two complex inheritance patterns are pleiotropy and polygenic inheritance. These kinds of inheritances sound a little bit the same, and so students often mix them up. So just make sure you take a little bit of time to understand them. Pleiotropy is the case where one gene impacts more than one character or trait inside of a human or any organism. So an example of this is sickle cell disease. Down here I have a picture of these puffy red beach balls. They carry oxygen so that we can do cellular respiration, so that we can make energy, ATP, for our cells. We've discussed this in previous lectures. Sickle cell is actually a single mutation, and that single mutation changes the shape of your red blood cell. And here, right here, we see this sickling shape, and this sickled shape is interesting. Even though it's one gene that has changed, it causes not just this one shape change, but a whole cascade of problems. So this one gene impacts more than one trait inside of us. Sickle cell disease can start with lethargy and weakness and things like that, but also pain and organ damage, so that organs start to fail all the way up until systems are failing for individuals. So this one gene impacts more than one trait because it's impacting this huge energy reaction that is so important for life. All right, the last complex inheritance pattern is polygenic inheritance. This is the case where we have two or more genes that impact a single phenotypic trait. I want you to imagine one of those old-fashioned balances that you can kind of tip one way or the other by adding certain kinds of things on either side of the balance. This is what's happening in polygenic inheritance often, or how I visualize it, is that more than two genes are weighing in on this final trait. And they're having an influence or a say in what that final trait is going to look like or appear like or behave like inside of an organism. Over here I have some examples. One of them is skin color. If skin color only had 8 genes involved, then you would have 64 possibilities in skin color. So it gets quite complicated quite quickly. But we think that there are about 378 color genes that influence that final skin color inside of humans. If we were still in class, I would ask you to look around at your classmates and you would see that you don't all look the same, right? Your skin colors are quite different, even from individual to individual. Within 100 individuals, we wouldn't have identical skin color types, right? And so this polygenic inheritance, again, these 378 regions and genes are like kind of weighing in on that final sort of color and that final appearance of that trait. Eye color, we think there are 16 genes that weigh in on that final gene. Height, we think there's 424 regions that weigh in on your final phenotypic sort of height that you reach in your lifespan. So I've started out this lecture, Variations on Mendel, to show you a few ways that Mendel, the laws of Mendel and what Mendel proposed gets to be more complicated as we step outside of that one inheritance pattern and explore some other ways that we add some complexity to inheritance. So I'll see you next time.